Okay. This session is dedicated for our module number 12. We'll have a few post-lab discussion notes for the activity on bacteriological analysis of water. Last time, we introduced the method of most probable number in order to estimate the number of cells that are present in our water samples. In addition, we also talked about coliforms. We discussed them under the light of these organisms being considered as contamination indicators, as fecal contamination indicators. So we also talked about the confirmatory and the completed tests, diba? Where we check on the growth of the microbes isolated from the water sample, whether they can ferment lactose, they can produce acid and gas, and when grown on plates, no, especially the selective differential plate, the selective differential medium, which is EMBA, we're able to appreciate how the colonies look like. No? So a few things that we'll cover for this session. No? Let's start with water quality parameters. I've discussed about how water quality is being tested. There are different parameters, physical, chemical, and microbiological. For the physical parameters, a few are listed here. How turbid the water is, what taste does it possess, how conductive the water is, the total dissolved solutes that are present, and other physical parameters. No? Chemical parameters such as pH, dissolved oxygen, residual free chlorine, radionuclides, and organic and inorganic chemical contents are also quantified in order to have a picture of how chemically contaminated potentially the water that we are processing is. In addition, we checked on the we also have to look at the microbial quality of water by doing coliform counting. Also, it's possible that we checked on specific bacteria, specific organisms of interest. And also for the case of parasites, we can also check on the presence of cysts or eggs of helminths that are present in the water. No? Those are actually microbial indicators as well. But the most popular one is coliforms, no? Yung ating detection and quantification of coliforms. So, I've discussed also that the quality of water will translate to whether it can be used as for drinking, or it can be used as for other purposes. No? Like if the quality is poor, normally or definitely we should not use it as drinking water, because drinking water is intended for human consumption and also for food preparation. Therefore, very important that the quality of intended drinking water is very high, that it doesn't have microbiological contamination, chemical contamination, and the physical parameters are acceptable. For potable water, no, we define potable water as something that is suitable for both drinking and cooking purposes. Though in some instances, no, yung drinking water class natin, it's much higher than potable water. Right now, I think potable water is something that we also have to check from time to time, kasi there are differences in terms of water treatment, no? Like how Maynilad and Manila water, how they do treatment might be different from our local water concessionaires in our provinces or our municipalities, no? Probably some of you are living in areas where potable or drinking water is supplied to you via a water concessionaire, a water district. So the processes that are employed for water treatment might be different. So it's important that we also have to check on the quality of potable water kung ano siya, no, talagang readily available for drinking purposes. Okay? The Department of Health, as well, in collaboration with the local government units of the different provinces and different regions in our country, they establish what we call a national standard for drinking water. No? There are few qualities, few parameters that we have to account in order to identify if the quality of water is acceptable for drinking. And a few are listed here. Microbiological content, heavy metal content, arsenic, cadmium, lead, nutrient content for the case of nitrate, chemical content, physical parameters, color, turbidity, and then other elements more or other chemical composition. No? Those are all included as parameters. Though lately, or it has been there has been a recent iteration of the national standards for drinking water, 2016, and they have elected mandatory, primary, and secondary parameters already. The list is very long, and I won't discuss that here. If you want, you can read through the Philippine National Standards for Drinking Water so you can see what things are being measured or accounted for. You know? And usually, the secondary parameters are the measurement of the secondary parameters. They're not done all throughout the water concessionaires, the water districts. No? Kasi yung secondary, ano lang siya, supplemental information. So, the LGU may or may not include it for water quality analysis. Okay? The most popular microbiological indicator are the coliforms. No? The microbiological indicator of water quality are coliforms. So we detect coliforms, we apply standard water analysis protocol, primarily yung ating sequence of of methods from presumptive to confirmatory and completed. So where we employ the multiple fermentation or the most probable number protocol. No? Aside from that standard method, meron din tayo membrane filtration, which I've discussed in our pre-lab also, can be used as an alternative to quantify coliforms or to check for the presence of coliforms. Now, the idea is that if we want to ensure the quality of water, the safety of water, in terms of the bacteria present in it, we can detect the certain group of bacteria called fecal coliforms because they tell us that, or their presence means that the water sample or the water source where it came from is definitely contaminated with fecal matter. Okay? What are the health consequences? Siyempre, if fecal coliforms are present in water and we consume water, that fecal contaminated water, diba? These pathogens may cause disease to us. And aside from disease, from infection, we may also serve as vehicles for these pathogens to increase their distribution in our environment. Okay? Now, some examples of coliforms are actually specific ones that are harboring our body, no, mga Enterobacteriaceae. Usually, we find them in our gut, in our intestinal organs. They are under the Enterobacteriaceae family. There are the lactose fermenting coliforms, and there are also those that are non-lactose fermenting enterobacters that are not essentially or usually not related as or identified as coliforms. No? So for lactose fermenters, some popular examples are the Escherichia coli, the Klebsiella, the Enterobacter, no? And if under the completed test, we visualize the colonies of these enterobacters using a variety of media. We can use McConkey, EMBA, SS agar, and we can also use triple sugar iron agar. That's another another example, no, of agar that we can use for the completed test. Pero for our case specifically, diba, we're gunning for the lactose fermenter. So more of the E. coli group, no, and its associated members. Okay, I've discussed this already. Yung characteristics of coliform organisms. We talked about in terms of cellular morphology. Most of them are rod-shaped and they are non-spore forming and they are all gram-negatives. They can ferment lactose at 37 degrees Celsius and you can expect that for under yung what do you call this? The confirmatory test. No, you can check on the production of acid and gas. Okay, and then also acetaldehyde is actually related to coliforms, the production of this compound. Okay, how do we check on the cellular morphology? Of course, we do gram staining, or we can check the growth of the coliforms in selected media. For lactose fermentation, we can check on gas production caught in the Durham tube dun sa confirmatory test natin. The colonies will usually have dark coloration, dark pink, red, purple, with green metallic sheen. Acid production is also identified or detected because there's yellow coloration in the lactose broth after the growth of the coliform organism. And metallic sheen and purple production, purple dye or coloration production with low in acidic conditions will be present. And then for acetaldehyde production, no, we can see the detection of, or we can detect it by appreciating dark coloration of the colonies when they are grown in appropriate selective medium. The fecal coliforms I've mentioned are positively correlated with fecal contamination. And most of the time, we associate them with us, with warm-blooded animals, okay? And these organisms, as rapid lactose fermenters, they have a range of cardinal temperatures, no, from 37 to 45 degrees Celsius. Okay, now let's review the method that we employed, which is the most probable number method. The most probable number method is a statistical method of determining microbial populations, a total coliform. And it uses a technique, or it is a technique that uses multiple dilution tubes, no, with the resulting number coding translated mathematically into tables. So you have your index values, your MPN per 100 ml values, no, and the MPN will indicate the presence or absence of an approximate or an estimate number of coliforms. So we employed this method under the presumptive test. No? So the presumptive test, yun nga, our water sample, we inoculated it into different tubes containing different volumes, no? Multiple tubes containing volumes of what do you call this, of the medium. So we have those that we inoculated with 10 ml, those with 1, and those with 0.1. No? After a while, no, we check on the positivity or the negativity under presumptive test. We expect that for negative, the absence of gas in broad tubes means that there's no coliform present. While for positive tubes naman, the Durham, you can see, no, magkakaroon siya ng space, gas bubbles that are trapped dun sa Durham tube natin. We expect that coliforms are potentially or presumptuously, we can presume na meron coliforms yung ating water sample. Okay? So yan, mention ko na ito. Okay. Yes, 10, 1 ml, 0.1. Okay, and then you can see, ito pala, no, one thing, sometimes when you grow or when you do the MPN, you do the presumptive method, sometimes it's problematic na hindi buong tube yung nagiging filled with gas. So that's possible. No? Normally, half will be sufficient to say that there is coliform. Pero if there's no like full production of gas, you can re-incubate your samples, your tubes, and check kung magpo-produce pa siya ng gas. Okay? For the water quality standards, there are thresholds that we employ. So according to the Philippine National Standards for Drinking Water, but this is actually 2007. No? Meron na tayong version on 2016. Total coliforms should be at the conformity risk level of less than 1 colony forming units per 100 ml for 95% of samples taken in a given time period. The detection of E. coli must give a result of less than 1.1 mpn per 100 ml. So very, very strict, no? Sobrang liit ng threshold natin for the standard of drinking water. So dapat ganyan yung values, no? An example, tingnan natin, no? You can check here. For an example, water from kitchen tap. Total coliform bacteria is identified as 50. No? Medyo malaki siya. So, this means that the water sample is not good. No? It's not potable kasi very, very high yung kanyang total coliform bacteria. Okay? Kasi according to here, yun nga, the total coliform bacteria must not exceed a value of 1, no? 1 colony forming unit or 1.1 mpn per 100 ml. Okay? In terms of E. coli, a few things or a few, range can be existing or interpretive, ranges. If less than 1, okay, not bad. 1 to 10 is low value. Intermediate is 10 to 100. High is 100 to 1,000. No? Isipin nyo may mga water samples where you can detect 1,000 colony-forming units of E. coli in 100 ml. Very high if it's already more than 1,000 colony-forming units. So as an example, let's have here a report from the DNR. Yeah. So here, they're looking at the change in the coliforms, fecal coliforms in different water sources or water sites before and after rehabilitation. So you can see from water sampling sites in Padre Faura, across Aristocrat and the Yak Club, no, where before rehabilitation, millions ang fecal coliform. After, bumaba naman, no, yung kanilang coliform count. Okay? Now, how do we determine that yung exact na MPN value per 100 ml? So, actually, that value is can be computed using a specific formula. So you just have to multiply the number of positive tubes with 100, and then you divide it with the square root of the product of the total ml of samples in the negative tubes multiplied by the total ml of sample in all tubes. So this is how you get the MPN index. No? This is how it is computed. So, pero may table naman, so you don't have to do it. But an important or a trivial information I want to share is this formula on how we get the MPN index per 100 ml. Okay? Next, we have the confirmatory test. So for the confirmatory test, we do this to confirm if coliform if what we have identified initially as coliform really ferments lactose. No? Kasi coliforms are lactose fermenters and they produce gas from it. What we use is brilliant green lactose bile broth. No? The contents of this medium facilitate yung ating confirmatory visual. Bile, which is present in the medium, is inhibitory to gram-positive microbes, while brilliant green dye inhibits selected gram-negative bacilli that are not really coliforms. So very important yung constituent ng dye tsaka ng bile. Of course, the lactose serves as the carbon source, definitely. And if the microbe, the coliform, yung ating presumptive coliform, is capable of really fermenting lactose, it will produce gas. Yung gas ay matatrap ulit doon sa Durham tube and makikita nyo siya as bubbles. So here, so positive that after the incubation period, Ayan, nagkaroon ng gas. So therefore, after the confirmatory test, sure tayo na yung presumptive coliform from the presumptive test is really a coliform. Okay, kung may gas. Kung wala, negative, hindi siya ngayon coliform. Then we do the completed test na to visualize the appearance of the colonies using selective media like EMB or less endo-agar. Okay? For the completed test, we use the selective differential medium such as aerosin methylene blue agar, and then we can check on the interpretation whether the coliforms are really present or not, or the coliforms based on their colonial appearance will really be identified as is. Let's take a look at some. Ganito yung itsura ng mga coliforms. No? Na ginromos sa aerosin methylene blue agar sa EMBA. So here you can see the we can expect that these colonies here are gram-negative rods and most likely coliform siya with metallic sheen, dark purple coloration. The contents of the medium also facilitate yung visual appearance. Methylene blue inhibits gram-positive bacteria. Aerosin changes color to a dark purple, especially when the medium becomes very acidic. Okay, so you know that acid production is key to coliform identification or key to the coliform group. So if the acid is produced, pH is decreased, the microenvironment of the medium becomes very acidic. Therefore, the organisms really a coliform. No, the organisms from the samples are really coliform. Okay, so here, a few things. Though, don't take this as a Bible. May ano siya, syempre may ano tayo, meron din tayong considerations pa. Usually, yung green metallic sheen in EMBA indicates E. coli. Dark pink, red can be Enterobacter, other Enterobacter species also. No metallic sheen. The pH did not really lower that much. Coliform, but we don't consider it as a fecal coliform, unlike E. coli, no? yung Enterobacter aerogenes. Then we also have other light pink, colorless non-fermenters, such as Pseudomonas. These are non-coliforms, no? Though in some instances kasi, we have lactose fermenters that appear pink initially pink sila. So baka mamaya, kung unang observation mo, sabihin mo na hindi siya coliform. As eventually, magiging dark pink siya o dark red. So, factor din yung time sa pag-observe ng expected results mo. Okay? So, here, some examples, visualizations of those. So, you expect, pagka negative talaga non-coliform, talagang almost colorless siya, no? There's no appreciable color ng colony. Parang very transparent compared to the coliforms, fecal and total. Okay, as an alternative, I also introduced the concept of membrane filtration. So it's a technique that uses a physical barrier, usually a porous membrane, to filter, separate particles and microorganisms that are present in water, so that we can trap them dun sa membrane, no? So the particles, microbes that are larger than the pore size will be captured by the membrane, by the nitrocellulose membrane. Okay? Major advantage of the membrane filtration method over the MPN method is that, of course, it isolates discrete colonies of bacteria, whereas in MPN, usually hindi natin nagagawa yun kasi we start with a very mixed sample. Also, bacteria trapped on the filter will grow as visible colonies already. Madaan na siya kasi ilalapag mo lang yung i-touch mo lang yung filter sa growth medium tapos mag-grow na yung, of course, kung ma-transfer yung cells, mag-grow yon as colonies, no? Like after the transfer, we can expect the growth, no, of the colonies. So some advantages of the membrane filtration technique, no, aside from the things I've mentioned already, mabilis siya, you can process very large sample volumes, no, because you can use only one filter for very, very large volume of water sample. The preparation time also for the membrane filtration technique is shorter compared to the multiple tube fermentation technique. Okay? No. So, example ng plate natin after membrane filtration. So, kita yung discrete colonies, no? That grew on the medium. Okay? So, that ends our session, our post-lab notes for module number 12.